Thank you very much for the introduction. I am OTAKI from Ryukyus University. Compared to Dr. Muso's research, our research might not be so much. I am not a radiology specialist, and I specialize in research of the patterns on the wings of the butterflies. So therefore, my achievement is completely incomparable to Professor Muso's, but I'd like to begin my presentation nevertheless. Target was the pale grass blue butterfly, which is a very small butterfly that is quite commonly seen in Japan, in rural as well as urban area in Japan. I would like to be very brief in my presentation because of the time limit. This is Naha because I am from Okinawa. And this is the most urban area of Naha, the Kokusai Avenue, which is the most populated urban area of Naha. And even in these areas, you can observe the pale grass blue butterfly in quite abundance. Why did we choose this particular species of butterfly? To give you more explanations, there are several reasons. First of all, the color pattern is easily distinguishable because it's a small size butterfly. It has a short life cycle, and it's easy to collect in the fields in various areas of Japan. And additionally, it is very fitted as an indicator species for environmental changes as well. Firstly, because they live on the surface of the ground, close to the ground. So it's easily perceived. The impacts of, for example, radioactivity on the ground is easily perceived, and it shares living space with humans. And it is found in most places in Japan, and it only eats katabami, which is oxalis, and that's the only thing that it eats. So it's easy to understand. It's monophagous. And we have experienced the March 11th, 2011 nuclear disaster, and we have come up with an idea that perhaps our experimental system and methodology can be useful to understand the impact of this accident. But that said, we are not a specialist in radiation. And the result of our research has been published in the scientific reports in the August of 2012. And as a result, there has been much response from the international scenes and media. This is Le Monde from French newspaper, which covered our study results quite extensively. However, that said, in Japan there was no report from the media and coverage of the media for a very long time. After a quite extended time, the Japanese media finally started to cover our research results. There are 4 sets of experiments within our research. First of all, the first part of our experiment is the field sampling that took place in May 2011, just 2 months after the accident. It's field sampling, collecting of butterflies, and the first Volten was developed. The same activity took place as second sampling in Fukushima in September of that same year, in 2011. And the fourth Volten was developed. And then the third point is the external exposure experiment. So we used the Okinawa larvae for the external exposure experiment on cesium-137. It's a very simple experiment on externally exposing to cesium-137 the larva from Okinawa, far away from Fukushima. The fourth and final experiment content is the experiment on internal exposure by feeding contaminated leaves of oxalis to the Okinawa larvae again. We collected these oxalis leaves from Fukushima contaminated area and fed them to a larvae that were in Okinawa, which were not exposed previously, to test the impact of internal exposure. The collection and sampling points were as follows: 10 areas, as you can see here. This is some pictures from the sampling activities of both the butterfly and the oxalis leaves. This is our activities of measuring radiation and radioactivity. The number of individuals used in the studies are as follows: 2,516 as F1 and 538 as F2, and again, in total 5,741 individuals. I'd like to share you the results of the experiment. The first experiment was first research was checking the wing size of the samples and the body size of the sample. And this is quite unexpected that there is a significant wing size reduction in the individuals from the Fukushima area. We have samples from Fukushima and Motomiya, and in these two areas, they each represent each individual butterflies. And you can see that compared to other areas, for example, Shiroishi area, which is north of Fukushima, or from Tsukuba or Tokyo, you can see that the wing size are considerably smaller, and there is more variation in these two contaminated areas. And if you plot this against the ground radiation dose, you can see clear causal effects as indicated here. As for the wing size, the results do not seem to follow the temperature size, because in cold areas, in cold climates, the individual's body size and wing size tends to be bigger. But that does not seem to be the case in this study results. Fukushima wing size should be larger, but that is not the case. It's getting smaller. And even in the areas north of Fukushima, like Shiroishi, you see the result as such. So we've collected this butterflies from many different localities throughout Japan. And so far, what we have understood is as such, but this is not a conclusive result yet. And we can see that there seems to be no geological decline observed so far. And the wing size seems to be considerably smaller in the Fukushima area. Wing size is almost constant throughout Japan, and no decline has been observed. We also studied the abnormality rate. You can see the green and pink bars. The green represents the May result, pink represents September results. This is the P generation, the latest generation of butterfly. As for the B graph, the F1. And the vertical axis, the abnormality, the horizontal axis in localities, the first one being Fukushima. And if you compare, you can see that the abnormality rate is increasing in all of the localities except Mito, which is the single and only exception. And you can see a similar trend for P as well as F1, that the September abnormality rate is higher. And in terms of the relationship with the latitude of the location, there is no conclusive decline in terms of the latitude. But as you can see, yellow dot, by the way, here represents the F1 and the new Fukushima Daiichi power plant. And we have also studied the F1 and days taken from egg to eclosion or hatching of the F1 generation. This is the eclosion curve. The horizontal axis being the eclosion time or days to hatching. Vertical axis, the percentage of the enclosed individual butterflies. You can see that 30% on this purple line has hatched after 21 days, and eventually all eggs hatch. And Tsukuba, the purple line, is sees quite early eclosions, whereas Fukushima eclosion time seems longer. The conclusion being that the closer to the nuclear power plant in Fukushima, the longer the time to hatch. There is growth retardation. Some examples of the abnormalities that we've observed in F1 generation: abnormalities in the legs, the wings, or the eyes area, and many different abnormalities in wings in various forms. Talking more about the abnormalities of the legs, we have mapped that the closer the distance to the Fukushima power plant, more abnormalities in the legs of butterflies. We also studied abnormality rate of F2 generation. We have the study of abnormal female from each locality crossed with normal tsukuba male. And what we wanted to study here is that in this F2 generation, there is relatively high abnormality rate, around 40-50%. So this shows that actually abnormalities are hereditary. And in case of abnormalities for leg, the abnormalities tend to be identical or similar to one generation. It could be slightly different type of abnormalities of legs or wings. So put together, there's about 70% abnormality rate in terms of similar abnormalities. So the conclusion here is that the abnormalities also are transmitted to the F2 generation as well. Now, this is quite a shocking result of how the antenna are the abnormalities are observed in the antenna as well. Normally they are straight, but this is not the case here, and they're split in multiple ways and various different ways of abnormality patterns in the wings. And we are not studying this in a DNA level. However, these results are not sufficient to demonstrate hereditary based on DNA damage. Now, also about the color pattern abnormalities in F1 and F2, the A section demonstrate normal wing types, and B section demonstrate aberrant or abnormal patterns. It might be difficult to understand the abnormality patterns, but since we have been focused on the pale grass blue butterfly extensively and for a long period of time, we can immediately detect abnormal patterns. So you would need to trust us when you say that we have been studying this type of butterfly for over 10 years, and in a normal conditions, we do not see abnormal patterns so frequently as you can see here in these areas. And diverse and unpredictable color pattern abnormalities often are associated with other morphological or behavioral abnormalities, such as abnormalities of legs, etc., and low reproductive performances. The color pattern, by the way, as I said before, have certain variations even among the normal wing types. So therefore, we have introduced a cold shock-induced phenotype study. So when you give cold shock, the temperature shock at the time of to the butterfly, you see this kind of impact to the wing patterns later on. So compared to this model, you can see that there are abnormal wing types from the high radiation area. Now about the external exposures, we have two different other studies. First of all, morphological abnormalities studying at 55 millisieverts and also 125 millisieverts. This is not a very low level of radiation. And with this external exposure, we have found that various abnormalities in legs, in antenna, in eye, copy, and wings. What is interesting here is the result of experiment measuring wing sizes. Compared to the control group, we saw that the irradiated butterflies tend to have smaller wings both in male and female. We have applied exact same method of experimentation. In terms of the control group and the irradiated group, it comes from the same parents. One irradiated externally and other not. As for the survival rate, we have created this line graph of survival rate, and you can see that for both 55 millisievert and 125 millisievert, you can see that compared to the respective control group, there are changes in the survival rate in terms of the decrease in survival rate. So therefore, the conclusion here is that there was a similar occurrence as what we have observed in the field data. Field data were reproduced. Finally, about internal exposure experiment. So in terms of methodology, we gave butterflies from Okinawa the oxalis leaves that were collected in the Fukushima area and also from Ube, Yamaguchi area that is far from the Fukushima power plant. We measured the concentration of radioactive cesium in the oxalis leaves or katabami leaves. And we studied the abnormalities, the wing size, and the survival rate of the butterflies that consumed these contaminated oxalis leaves. The collection sites of the oxalis katabami are, as you can see. So Ube, Yamaguchi, very far from the Fukushima area in southwest, Hirono, Itate, and Fukushima. And these are the data array of leaves collected from different localities. However, this is difficult to see, so I have reconfigured the chart to set Ube level of radioactivity as 1, and compared to that you can see the difference in radioactivity. What are the abnormalities observed in terms of impact of internal exposure? Abnormalities in antenna, palpi, eyes, and wings are again observed in various different forms. As for the survival curve, in Ube, those that were fed the oxalis leaves from Ube, Yamaguchi Prefecture, were all survivors. They all survived. 100% survival rate. Those who consumed Itate area leaves have a certain decline in survival rate, as well as Hirono. This other chart shows the four-wing size, comparing Ube on the very left-hand side with other areas, and you can see that there are some discrepancies in the wing size as well. So again, our conclusion is that the field data was again reproduced. Now I'd like to summarize the results of our findings. There was growth retardation and small forewings observed. There were morphological abnormalities. There were increasing abnormality rates in all generations we've studied, and inheritance of abnormalities were observed as well, and other findings regarding external and internal irradiation. And the important points and unique points of the studies are as follows. First of all, we started the monitoring immediately after the March 11th accident, which was quite meaningful, immediately after in May 2011. We also evaluated by rearing F1, F2 in Okinawa, which is a completely contaminant-free environment in terms of radioactivity. And thirdly, reproduction of the field and rearing results by external and internal exposures were undertaken. Our study is about long-term low-dose exposure, which is different from short-term high-dose exposure, and there are relatively small amount of information regarding the low-dose exposure in this area. So therefore, I believe that is another meaningful point of our study. We are also monitoring the adult abnormality rate of these butterflies in the course of time. This data is currently unpublished, but if you look at the data in May 2011, the abnormality rate is relatively low. We are not seeing so much symptoms, but in September we have sudden increase and hike in the abnormality rate. There has been several generations of these butterflies that are hatched in between May and September time, and then we have seen some decline since the September 2011, and this is due to generational change of the pale grass blue butterfly. This concludes my presentation. Our study is undertaken by a lab in Ryukyus University with these members. Iwasaki-san, Iwata-san, Taira-san, Kenjo-san, Nohara-san, and Hiyama-san. We have these participants and collaborators in our research at Ryukyus University and many other people. Thank you very much for your attention. Professor Otaki, thank you very much. I have a question to Professor Muso. Do you have any comment on this presentation by Professor Otaki?
First, I think it's a wonderful study. It's an incredibly comprehensive, scientifically rigorous study. It's exactly what I would do if I lived in Japan.
まず最初に大変包括的で、そして科学的にも徹底した調査であるということで素晴らしい研究内容だと思いました。私も日本に住んでいたらこのような研究を行っていたと思います。
My main comment is I hope that you will succeed in acquiring additional grant funding to continue your studies at a broader scale, perhaps with other species as well. I think this is very important that this kind of work continue.
このような研究活動というのが続けられるということが大変重要と考えますので、今後新たな資金提供を受けまして、よりその研究の範囲を広げられて、他の種にも広範囲に広げられて継続されるということを希望いたします。